puck now, down low for Gaudreau to Kachuk in the corner, back to Johnny Gaudreau. Gaudreau in front, feeds a pass to Lino, stopped by Ottinger, took it up high. Gaudreau scores!
In Canada, ice rinks are more than just recreational spaces. They are a cornerstone of our culture and identity. From backyard rinks to international arenas, they are where future hockey legends are born, and where our nation's passion for winter sports is celebrated. They are a form of cultural heritage, a symbol of our resilience, skill, and the strength of our communities. These rinks are woven into the very fabric of what it means to be Canadian, but maintaining them comes with a growing environmental cost. Most rinks rely on large-scale refrigeration systems, where compressors circulate chilled brine beneath the rink floor to keep the surface frozen. Yet, with heat constantly seeping in from the surrounding environment, maintaining the low temperatures for ice to freeze between -4 and -8 degrees Celsius takes serious energy.
Take the Olympic Oval in Calgary, known for having the fastest ice in the world. It's one of Canada's best facilities, and it spends over $45,000 a month on refrigeration alone. And while the Oval can afford it, most local rinks can't. Keeping ice frozen consumes over 520 kilowatt hours of energy a day, equivalent to what a household consumes across an entire month. Spread across year-long operations in over 3,000 rinks nationwide, ice rinks are the largest energy consumers in municipal areas, accounting for 40% of corporate building emissions. The result? Rising operating costs, increased carbon emissions, and the loss of a beloved tradition carved into ice.
Several alternatives have been proposed, though none fully address both sustainability and performance. Synthetic ice made from polymers reduces energy used by eliminating the need for refrigeration. However, these polymers cause blade dulling and restrict movement, making them unsuitable for high-performance activities. Greener refrigeration methods have also faced limitations. Ammonia and carbon dioxide systems, though environmentally friendly, require high installation costs and are expensive to implement, making them unfeasible for community rinks. As the problem remains unresolved, it is crucial to develop a sustainable, long-term solution that reduces energy consumption while preserving the skating experience.
We are RHS-Calgary, a high school team addressing the energy-intensive nature of ice rinks with an innovative and sustainable solution. Our approach utilizes ice-nucleating proteins, or INPs, which promote ice formation at higher temperatures. By applying INPs in ice rinks, we can maintain surface quality without keeping the arena at excessively low temperatures, reducing the energy required for cooling. Our research has 3 specific objectives.
First, to transform E. coli with the INA-Z genes from Pseudomonas syringae to produce and purify ice-nucleating proteins.
Second, to run molecular dynamic simulations on INP variants to determine the most effective protein construct.
Third, to test our purified proteins in the lab and compare their performance against commercial INPs.
We hope this project will encourage ice rinks to reduce their reliance on cooling systems, promoting environmental and economic sustainability. This approach aligns with UN Sustainable Development Goals 12, Responsible Consumption and Production, and 9, Industry, Innovation, and Infrastructure.
Our wet lab team aims to transform E. coli with a plasmid encoding for ice-nucleating proteins, INPs, native to the pathogen Pseudomonas syringae. INPs consist of 2 main structures, the WO and R coils. The WO coils form the majority of the protein and feature repeating threonine X threonine motifs, where X refers to any amino acid, which fold into a beta helical structure. These motifs contain both hydrophilic and hydrophobic groups, as represented. The hydrophilic groups, marked by the blue arrows, attract water molecules, forming the active site for ice nucleation. Meanwhile, the hydrophobic groups, shown in white, trap water between the loops of the helix, ensuring stability. The structure of the protein itself resembles that of ice crystals, ensuring water nucleates around the protein. The second part of the protein, the R coils, contains repeating motifs that play a crucial role in ice nucleation and protein structure. Studies have shown that reducing the number of motifs hinders ice nucleation, while adding motifs may enhance it. At the end of the R coils, the C terminus is responsible for protein folding, tertiary structure, and facilitating ice nucleation. On the opposite side, the N terminus controls environmental responses and anchors the proteins to the cell membrane. A key characteristic of IMPs is aggregation, where proteins chain together to increase surface area for nucleation. In ice rinks, water begins to nucleate between -4 degrees Celsius and -8 degrees Celsius. Yet IMPs can promote nucleation at around -2 degrees Celsius, providing a promising approach to reduce energy costs in ice rinks.
We plan to transform E. coli with INAZ gene, which encodes for IMPs. At last year's GOGEG conference, we proposed using molecular cloning, MoClo, to assemble our plasmid. MoClo is a genetic engineering technique that uses type 2S restriction enzymes and standardized overhangs to assemble multiple genetic parts into a single plasmid in a defined order. It is widely used in synthetic biology because it enables efficient multi-gene assembly, standardization of parts, and easy swapping or reuse of genetic components. However, as our project developed, we identified a major limitation with this approach. The INAZ gene contains many repeating sequences, which increase the risk of assembly errors when using MoClo. We explored splitting the gene into smaller fragments, but this approach would still be prone to error. As a result, we chose to move away from MoClo and instead use a more traditional cloning approach with previously published plasmids, which were successfully expressed and purified in E. coli. This offers a greater chance of protein purification success when working with modified versions of IMPs.
Han et al. Determined that the most effective ice nucleating constructs were INAZ-240 and INAZ-576, which contain 15 and 36 repeating motifs. We selected these two variants for several reasons. First, both constructs showed high elution volumes, indicating more orderly protein structures. Second, INAZ-240 is smaller and more soluble, making it easier to purify, while INAZ-576 formed thicker bands in the SDS page, suggesting greater expression, aggregation, and potentially higher activity. Importantly, previous studies showed that aggregation only improves efficiency up to a point, as variants that aggregated more than INAZ-576 showed reduced performance. Finally, when aggregation data was analyzed, both INAZ-240 and INAZ-576 produced high, symmetrical peaks, unlike other variants that showed shouldered peaks indicative of unstable structures. This homogeneity suggests that these two plasmids yield high concentrations of effective proteins. As such, we plan to transform E. coli with both plasmids, while designing alternate versions of INPs with modifications to increase nucleation efficiency and protein yield. Firstly, we aim to truncate the N-terminus, which anchors the protein to the bacterial surface. Previous studies have indicated that anchorage reduces the effective surface area for ice nucleation. By removing this region, we aim to allow E. coli to produce the protein intracellularly, facilitating purification and increasing activity. Secondly, we aim to manipulate the number of repeating motifs within the protein. Han et al. Determined that the ice nucleating proteins are most effective between 6 and 39 repeating units. Too few reduces activity, while too many prevent aggregation, which is essential to form structured ice lattices. By analyzing previously studied structures and using molecular dynamics simulations, we will construct variants with differing numbers of motifs, aiming to identify the optimal protein structure for ice nucleation.
So far, we have successfully made new batches of competent DH5 alpha strands of E. coli, which were transformed with GFP, confirming that our cells are competent to take up our gene of interest. We are currently performing mini preps to amplify our plasmids. In the coming weeks, we plan to transform the INAZ plasmids into DH5 alpha and BL21, DE3 cells, followed by a mini prep, gel electrophoresis, and a protein purification to isolate the protein for future activity assays.
Our dry lab team has two primary objectives. First, to model interactions between IMPs and water to understand the mechanism of ice nucleation and compare protein variants. Second, in line with Wet Lab's goal, to compare the efficiency of our protein's nucleation to commercial IMPs in the lab. We've begun to run molecular dynamics or MD simulations using GROMACS, an MD simulation package. At last year's conference, our team began introductory tutorials, such as modeling lysosomes in water, to build foundational skills. However, due to limited processing power in our high school lab, we were unable to run complex simulations. To overcome this challenge, we collaborated with Dr. Elio Sino at the University of Calgary, who assisted us in modeling a shortened IMP construct depicted here. The modeled protein contains 16 repeating TXD motifs, which are known to play a central role in ice nucleation. We selected 16 repeats to ensure that nucleation would occur, while keeping the protein small enough to model. Additionally, modeling programs favor even numbers of repeating units for symmetry, making this construct well suited as an introductory model. This video shows the MD simulation results. Water molecules are depicted in red, surrounding the ice nucleating protein in blue. Over 5 microseconds, we observed the formation of approximately 600 new water-water hydrogen bonds around the coils of the protein. These bonds are organized into a hexagonal ice lattice, shown in yellow. Quantitative analysis shows that water density increased over time, consistent with the formation of an ice lattice. Simultaneously, the number of hydrogen bonds increased and the system enthalpy decreased, indicating the formation of ice. Together, these trends provide molecular-level evidence for how IMPs initiate ice nucleation. This year, we gained access to a high-performance computer and have begun independently running GROMACS simulations. To date, we have successfully completed tutorial simulations of lysosomes in water to build proficiency with the software. Moving forward, our next steps are to complete the remaining GROMACS tutorials, simulate both INAZ 576 and INAZ 240 proteins, and then model the proposed N-terminal modifications and changes in motif numbers to determine the best variant. These results will directly inform Wet Lab's design and develop a more effective IMP.
To establish a performance benchmark for IMPs, we conducted experiments using commercial proteins, specifically SNOMAX. SNOMAX is a commercially available snow-inducing agent derived from Pseudomonas syringae and is widely used in ski resorts to form snow at higher temperatures. However, it has not been implemented in an ice rink. SNOMAX consists of lysed and lyophilized P. syringae cells containing INPs. In contrast, our project aims to improve nucleation efficiency by purifying the proteins, rather than leaving them embedded in lysed cells. We hypothesize that isolating the protein will increase surface area for reactions, increase nucleation efficiency, and reduce the concentration required for ice formation. We performed controlled experiments to determine the minimum concentration and temperature at which SNOMAX induced ice formation. Initial tests last year used large 3D printed rink models. However, these systems could not reliably reach sub-zero temperatures and were therefore unsuitable for experimentation. As a result, we redesigned the experiment using small volume serial dilutions, where various SNOMAX concentrations were compared against a control of distilled water. Samples were placed in a microcentrifuge tube rack and submerged in a cooling bath composed of ice, water, and rock salt, allowing sub-zero temperatures to be met. The bath was monitored over 3 to 5 hours, with both temperature and state of freezing recorded at regular intervals. Over 10 trials detailed in our paper, we identified that 0.25 grams per liter is the lowest concentration at which SNOMAX consistently induced freezing at -2 degrees Celsius. Importantly, all distilled water controls remained liquid under identical conditions, attributing ice formation specifically to the presence of INPs. As Wet Lab produces recombinant INPs, we aim to repeat this experiment using both SNOMAX and our purified INPs to compare nucleation efficiency, where we expect our proteins to function at lower concentrations and higher temperatures. In the future, we aim to return to the scaled rink models to determine if INPs can form ice sheets.
To promote community sustainability, our human practices team engages directly with ice technicians, athletes, industry stakeholders, and the public to assess project feasibility and raise awareness about the environmental cost of ice rinks. A key stakeholder in our research was Mark Messer, director of the Olympic Oval and world-renowned ice specialist. Our team is given an exclusive tour of the Olympic Oval's refrigeration system, and we learned that surface temperatures are kept at approximately -15 degrees Celsius for over 9 months each year. Messer explained that refrigeration alone accounts for 60 to 70% of the facility's electricity and water usage, costing roughly $45,000 per month. Messer also highlighted the aging infrastructure, pointing out leaks in refrigeration system and describing current maintenance efforts as putting band-aids on top of duct tape. In an interview, former Olympic speed skater Gaëtan Boucher echoed these concerns, stating that the Oval's ice has become slower due to poor refrigeration. When discussing our project, Messer expressed strong support for finding sustainable ways to save the rinks, but raised concerns about whether adding proteins could affect the ice speed and quality. Crucially, this interview shifted our focus towards community ice rinks, which face the greatest financial risks due to limited funding, but do not require the extreme performance standards of elite arenas. As such, we've begun to reach out to local rinks to explore practical implementations of our research.
Last year, our team attended the ISU Speed Skating World Cup and spoke with spectators, former Olympians, and event volunteers about rink sustainability. To our surprise, many were unaware of the environmental and economic costs of refrigeration. Yet, once we explained our project solution, they were supportive and recognized the importance of implementing energy-efficient practices. Skating experts shared stories of rinks that had to be shut down because of melting ice, emphasizing how the application of INPs can be invaluable for rinks struggling to produce cold environments. These conversations showed strong public interest in sustainable ice solutions, while also revealing a lack of public knowledge on the environmental challenges in these facilities. To address this knowledge gap, our team ran workshops for grade 4 and 5 students at our school. Students were challenged to design small insulating devices to keep ice from melting. As they quickly discovered, keeping ice frozen is surprisingly difficult, just like in real ice rinks, where massive amounts of energy are required. We shared key statistics and costs, and many students related them to personal experiences in local arenas. This activity helps students understand the environmental impact of ice maintenance, while also sparking interest in synthetic biology and encouraging creative problem solving as our project develops. We aim to continue running educational workshops, connecting with stakeholders in community rinks, and developing a website to engage the broader public with this issue.
Ice skating is a tradition that has endured for decades, providing Canadians with a sense of pride, joy, and connection to our culture. Without it, we risk losing a vital link to our national identity. Now, more than ever, it is crucial that we take action to ensure the sustainability of these beloved spaces, using innovation and community outreach to preserve this cherished sport for future generations. Welcome to Innoskate.